My name is Vivianne Tawfik, and I'm an assistant professor at Stanford University Department of Anesthesiology, Perioperative and Pain Medicine. I want to thank FAIR and the MSARF program for inviting me to speak today and share my career trajectory. I thought I would first start with a slight overview of my career so far, and then go into details of this path and provide some unsolicited advice. I started college, as you call it in the United States, but actually I went to university in Canada and did a 4-year biochemistry program. This was followed by an MD PhD at Dartmouth College in New Hampshire, which was a 7-year combined program, followed by an internship in general surgery for 1 year, also at Dartmouth. And then I moved all the way out to California for my residency in anesthesiology as part of the research track, which was 3 years. After that, I did a 1-year research postdoc with Gregory Scherer, doing really truly basic science, looking at molecular mechanisms of pain and control by opioids. I then went back and did a clinical medicine fellowship, clinical pain medicine for 1 year, followed by another research post 2 years with David Clark, also at Stanford, and then eventually was appointed as an assistant, but wasn't given any lab space. These are the 3 on me and worked in my lab, which was actually just a desk that we shared with 3 people, and it was a little bit, you can tell. And then finally, about 2 years ago, I was given training. In addition to that, I had a side project and had 2 children along the way, Gabriel, the little one, and Nathaniel, the big guy, both in their. So the first point of advice I wanted to give is that you should really look for a supportive mentor who's passionate about the science they do and truly sees their success as your success. And that's what I found at Dartmouth in Joyce DeLeo, who is a basic science researcher who I was lucky enough to work with for my thesis. At the time, she was really interested in spinal cord glia, how they're involved in pain. And because she was so excited about this project, I became so excited about this project too. And years later, as you can see, this is a review authored by a current MSARF student, Elena Haight, as well as me. The neurons are the only cells involved in pain. And we're really interested still in how astrocytes and microglia are involved in pain generation. During my PhD, this is just one piece of data from that time, I used a glial modulator and shown that after injury in a mouse model, a glial modulator can actually decrease the amount of pain or injury type responses seen in mice. And you can see here that it drops down precipitously when given the glial modulator. We published this and I pretty much thought I had solved pain, which is obviously not the case. And of course, there's still much work to be done. When I graduated from the MD PhD program at Dartmouth, I joined what's called the Fellowship in Anesthesia Research and Medicine at Stanford, as we refer to the FARM program. And what FARM is a physician scientist training program, or PSTP. And these exist at multiple different institutions for multiple different specialties. And they're really residency tracks that are developed for those interested in a career in academic anesthesiology and research. They allow up to 9 months of research during residency, followed by a 1-year research fellowship, or as you can tell in my case, many years of research fellowship to follow. And during that time, you're attending in the operating room during your clinical hours. So I joined this PSTP program, the FARM program, and worked with Gregory Scherer, and was really interested in studying pain still, but also interested in studying opioids, as this was really his area of expertise. I applied to FAIR, and you'll see that this is a theme here. I applied to FAIR for their Research Fellowship Grant, or the RFG, in 2013 with a project entitled Opioid Signaling in CNS Glia, Implications for Opioid Analgesia Intolerance. This was really a combined interest for Greg Scherer and myself, where we could bring glia, my love, in with opioids, his interest. And at the time of this application, I put together this career development plan. I would do 6 months of research during residency, then graduate, do a 1-year research fellowship, then move on to my 1-year clinical pain fellowship, and then the future. So this is where we were. I applied for the FAIR RFG, and I was lucky enough to have it awarded. And this was truly exciting for me and was really my beginning the beginning of my interaction with FAIR. Now, one problem came up when I joined Greg's lab. He was doing some of the most interesting and advanced science, and I had been working clinically for years between my finishing medical school, my internship, and my residency. And during that time, the interest in glia and pain had really increased exponentially. And this is just looking at number of publications using those search terms. And you can see here, this is when I started my PhD in 2002. There weren't that many people interested in glia and pain. By the time I got back to research after my PhD and after all my other training, people were definitely increasing in their interest in pain and research, in glia and research. And so this became a bit of a difficulty for me. So my second point of advice is that science will move on without you, but you can jump back in with a good mentor, protected time, and a willingness to learn. And in my case, it was sort of like starting over. A lot had happened in the glial field, a lot had happened in pain research, but a lot had happened in science in general with the development of Cre-Lox systems, transgenic mice, CRISPR-Cas9. All of these things had happened while I was busy caring for patients. But that's okay, and you can get back into it. So getting back into it for me meant collaborating with some wonderful postdocs and graduate students in Greg's lab, and we were able to put together a really great story about the role of mu-opioid receptors in neurons, not astrocytes or microglia. And this was sort of the culmination of that work, where I found that microglial cells, shown in this kind of yellow color, don't express the mu-opioid receptor, which is shown in this reddish color in the background, but actually it's spinal cord neurons and primary afferent neurons that express the mu-opioid receptor. So fast forward, I then went back to the clinic. I spent a year almost entirely doing clinical work as a pain fellow, and decided that I would really focus on that for the year. I learned a lot about chronic pain and had the opportunity to work with some really incredible clinicians at Stanford, and really started to become interested in the mechanisms underlying different types of pain. In the clinic, I was exposed to a condition I had never seen before called complex regional pain syndrome, or CRPS. And this was particularly interesting to me because it was a type of pain that occurred after or surgery, and patients ended up with red hot swollen limbs and decreased ability to move those limbs. And I couldn't really understand what was going on, and became fascinated with trying to figure out what I could do to understand better the mechanisms underlying CRPS. So I started to think about how we could model CRPS in a rodent, because all my research to date had been in mouse models. Luckily for me, I found that David Clark, who is another physician scientist at our Stanford VA hospital, was also interested in CRPS and had developed a mouse model for this disease. So I decided that after my clinical pain fellowship, I would go back and join Dave's lab, where I would learn the CRPS model and start to investigate the mechanisms. So again, I went back to FAIR, and this time I went with the Mentored Research Training Grant, or MRTG, and applied for it with a project entitled Monitoring and Modulating Microglial Cell Activation in Pain. The plan was to come out of the clinical pain fellowship and go on to either this grant or a K award. I was awarded the FAIR MRTG, which was very exciting. At the time, I also applied for an IARS Mentored Research Award, but I withdrew from that one when the FAIR was awarded. And I applied for an NINDS K Award, which was scored but not funded. So that brings me to my next point of advice, which is you should cry a little but not a lot when your grant is rejected, your paper is rejected, or you just generally feel rejected. And I just thought it would be fun to put in some of these rejections I've received over time. These are for manuscripts. Dear Dr. Tawfik, thanks for submitting your manuscript. Unfortunately, we thought it was terrible. Dear Dr. Tawfik, thank you for considering our journal for publishing your manuscript. Unfortunately, the reviewers thought it was not worth publishing. And then this one where they didn't even say Dr. Tawfik, because apparently I was just Vivianne at this point. Thank you for applying for this grant. There was such a talented pool that we didn't select you. So of course, even though there's some wins in between, there also are a lot of rejections. So following the rejection of the K Award, but the awarding of the FAIR grant, I was able to rethink the K08 submission and put it together again as a grant entitled Peripheral and Central Immune Contributions to Pain Chronification. And I was awarded this grant, so that was very exciting. And now I'm in kind of years 3 and 4. And again, this is sort of my plan that I had put together and said that the things that I would do in the future when I applied for this grant 4 years ago. And so we are now in the process of writing up some of our results, preparing an R01 for submission this fall, and analyzing data and growing my team. So all that said, it ends up being a bit of a long path of academic medicine from undergrad, med school, residency, fellowship, and then hopefully eventually faculty. But it is totally worthwhile. And I'll just highlight that by saying that I truly now have the best job in the world. I spend 70% of my time on research. I spend time asking interesting questions about chronic pain mechanisms, looking to discover new mechanisms, using my clinical knowledge to ask more relevant questions. I get to mentor trainees, and I actually have my first graduate student in my lab right now. I get to interact with colleagues from around the world. My research also influences my clinical work. I spend 25% of my time caring for patients with chronic pain. I get to bridge my research knowledge into practice. I get to mentor trainees, residents, and fellows. And I get to work closely with doctors and clinicians to care for complicated patients. Through both of these experiences, I also get to interact with colleagues from around the world at conferences and through seminars. And then I also spend some amount of time teaching and working administratively, running the farm program that I was a part of, our PSTP at Stanford. So when I say take my advice with a grain of salt, what I mean is that you should take all the advice you can get, but internalize it, process it, and place it in the context of your life, your passions, and your ideal career. As Deb Schwinn and others have written about our field, the future of anesthesiology is dependent on the success of young investigators who develop independent careers in science. And it's really a leading priority of our field to nurture the academic careers of physician scientists, your careers, to become part of our specialty. And just to show you, residency field chosen by MD PhD graduates, and of course not all of you will have PhDs, and nor do you need to, but when they looked at the fields chosen by MD PhD graduates, anesthesiology was showing up here at just 2.9%, pain medicine at just 0.2%. So the way I figure is you can go into internal medicine as a physician scientist and be a small fish in a big pond, or you can come to join us and be a big fish in a small pond, and we really do need people who are passionate about research and clinical work. So you've made it this far now. I think it's important to realize that what people see is not really the whole story. And there's a lot of persistence, failure, sacrifice, disappointment, and hard work that goes into it. So where does it leave us? I want you to get involved in research. If it's something you're passionate about, find mentors who can support your career trajectory. Consider joining a PSTP or a T32 program to help you navigate the system after you graduate from medical school. And a final plug for early stage anesthesiology scholars, which is a group we formed about 5 years ago now to support people like you who are interested in physician scientist careers in academic anesthesiology. Thank you so much.